So this was an image made by a woman in our department of a bacteria in the picture of King Kong being killed by the compound that we found and elucidated here in this paper. So the title of my talk and the title of my paper is that a dual mechanism antibiotic can target both gram-positive and gram-negative bacteria while avoiding drug resistance. And so as the wonderful intro videos that were showed while we were waiting for people to join described, if we don't do anything about antimicrobial resistant infections, oh, sorry about that, over 10 million deaths could occur from those by 2024, which is even more than cancer. And one of the reasons this is an issue is that because during the traditional antibiotic discovery pipeline, where you identify a molecule, you test it to identify which one is going to be the most effective, you figure out how it works, and then you put it into the clinic. This process over the past 20 to 30 years has only resulted in 9 new antibiotics, none of which have a unique method of killing bacteria. They're mostly just derivatives and recapitulations of old antibiotics that we've been using for the past century. So this is the bottleneck. We're discovering new antibiotics, determining how they work. This process is usually done by screening different organisms to find ones that are resistant. However, that's both time insensitive and not cost efficient, but it's also counterintuitive. You're looking for an antibiotic that's not going to be prone to drug resistance, but the only way to learn how it works is by identifying mutants that did become resistant. And so in my project, we had this question: how do we identify antibiotics with unique and novel mechanisms of action? And the way that we did this is using a process called bacterial cytological profiling, or as I like to call it, bacterial autopsies, where if you see a crime show, you can identify how that person was killed by the way the bullet entered the wound, what caliber the gun was by identifying what kind of shell came out. All of these clues you can gain from identifying just what happened to that body. And in the same way, using fluorescent dyes, you can actually stain bacteria. And we found, that different classes of antibiotics killed bacteria in a very distinct but similar way. And so if you killed a bunch of bacteria with, let's say in this case, 5 different antibiotics that all disrupted the structural integrity of the bacteria by attacking their cell membrane, they're all going to cluster together. And the reason they cluster together is because they all look very similar to one another under the microscope. So we did this for a large library of compounds, and we found one compound, SCH 79797, which I'll just be calling SCH from now on, that looked distinct. It did not look anything like any of the other antibiotics that we tested. And that can be seen by the distance and the size of this blue line up here at the top. So just like in previous experience, we wanted to see if we could identify how this molecule worked by identifying mutants that were resistant. And so as a control, we use several other antibiotics that are used in the clinic. For example, this molecule, trimethoprim, over the course of 30 days, acquires a high level of resistance in MRSA. Novabiocin, which acts in a different way to trimethoprim, sees the same high levels of resistance. However, SCH, this molecule that we found, did not become resistant. No matter how long we did it, or no matter what organism we used, we never were able to acquire resistance. And not only were we not able to acquire resistance, but this molecule was able to kill both gram-positive bacteria, which are listed in black, and gram-negative bacteria, which are listed in red. And while all of this is exciting, it doesn't really matter if we can't kill bacteria inside of an in vivo system. And so work done by my partner, Gabriel Moore, we actually used a in vivo worm model of bacterial infections and found that when you treated these worms with dangerous bacteria and SCH, their survival in purple was actually extended. However, if you left them alone or used other antibiotics that are used in the clinic, they still died at the same rate. So this is incredibly exciting. We have a molecule that is not only powerful, but bacteria can't become resistant to it. And it works both in the lab and in an animal model. But while this is so exciting, this is also kind of the problem. While it's exciting to not be able to get resistance, sorry, that is the main way that we identify the mechanism of action. So we had to get a little bit creative. And one of the ways we did that was using this method called thermoproteome profiling, along with a collaborator over in Germany. And what this process does is it uses mass spectrometry to measure how much of the protein inside of a cell remains stable and in solution as you increase the temperature. And we've shown that as you increase the temperature, certain proteins are more stable and they'll stay in solution for longer, like the orange ones or the ones to the far right, but others like the purple will crash out of solution very quickly. But what's interesting is that if you add a drug that binds to any one of these particular enzymes, you can actually increase its thermal stability. So we did this experiment on all of the proteins that can fit inside of an aqueous solution inside of the E. coli cell, and we found that there was only one protein that was stabilized. This protein is called DHFR, or dihydrofolate reductase. This molecule is responsible for many important metabolic functions, from DNA synthesis, protein synthesis, and amino acid synthesis. However, there's another antibiotic called trimethoprim that also targets the same enzyme. And if we do the same assay with trimethoprim, we see that DHFR also binds to this molecule. So we wanted to confirm that while it binds, does it actually inhibit dihydrofolate like we see trimethoprim do? And so using mass spectrometry again, we did metabolomics and measured of the level of metabolites that are listed here in various colors. And as you can see listed on the left, only one metabolite increases in concentration after treating with SCH, and that's dihydrofolate. And this suggested to us that kind of like a dam, there was a blockage here, meaning that any metabolite that was made above this point, sorry about that, any metabolite that was made above this point increased in concentration, while anything below it got rapidly eaten up and was never replenished. So this convinced us that yes, SCH is not only binding to dihydrofolate reductase, but it's also inhibiting it. But I thought that our initial experiments suggested that SCH was in a league of its own, that it was unique. And if I pull back up the data that I showed you in the beginning, you can see that SCH is all the way up at the top, separate from everything else, and including it's separate far away from trimethoprim, this molecule that it shares a mechanism with. And so we went back to the data, which you often have to do. And after staring at it for a while, I noticed that there were some bacteria that when treated with SCH became permeabilized. And you can tell because they have this green fluorescence present. This green is only present when the cell membrane has been compromised. And this is something that trimethoprim does not do. And so we asked the question directly, does SCH target the bacterial membrane? So to do this, we use flow cytometry, where you can actually measure the fluorescence intensity of a single bacteria. And we were able to measure the fluorescence of two different dyes that tell us two different things. One dye communicates to us the membrane potential of the cell, and the membrane potential of a bacteria is the charge difference across the membrane that's responsible for producing ATP and energy. The inside of the cell is negatively charged and the outside is positive. The membrane permeability refers to the ability of the cell to keep out big things. And so this dye only gets into the cell if the membrane has been compromised. And so if cells appear here in this bottom right-hand quadrant, they're healthy. They're making energy because they're polarized and they're impermeable. Nothing harmful can get in. However, if they're in the first quadrant, they're depolarized, which means they can't make energy, and they're permeable, meaning things can get in. If they're in the second quadrant, they are not permeable, but they are depolarized. And so if you treat them with a vehicle control, DMSO, which is the solvent that we dissolve our drug in, you can see that 95% of those cells are healthy and only 1% are unhealthy. And so what happens if we treat with a known control? CCCP is a molecule that is known to depolarize bacterial membranes and prevent them from making ATP and energy. And as you can see, when you treat drugs or treat bacteria with CCCP, 99% of the cells are depolarized. And if you treat them with a comparable control, measuring whether they're permeable, like niacin, you can see that while some of the cells are still healthy, a large majority of them are actually not only depolarized, but also permeable. And so we wanted to do this experiment with our drug. First, we tested the control, which is trimethoprim. This molecule shares the mechanism of folate inhibition with SCH, but we wanted to see if it was distinct in terms of helping bacteria keep their membrane integrity. So when you treat bacteria with trimethoprim, over 90% of those cells are perfectly fine, even though they are being killed and their folate metabolism is being hindered. However, if you treat these same bacteria with SCH, you see a completely different story. You see that these cells are not healthy, they are not polarized anymore, and they are permeable now to dangerous molecules like SCH. This suggested to us that SCH is truly unique. However, it's not unique because there's only one mechanism of action that's unique. It's unique because there are two that, when combined, make this molecule really special. And to test this, we went back to our first assay, these bacterial autopsies, suggesting that, or hypothesizing that while trimethoprim or a membrane disruptor like niacin alone won't create a bacteria that looks similar to SCH, if you combine trimethoprim and niacin together, just like SCH, the bacteria might look similar. And that's in fact what we saw, that if you look at bacteria treated with a vehicle control that shouldn't hurt them, trimethoprim, niacin, and the combination of the two, it is only the combination of the two that makes the bacteria look similar to SCH, not either of them alone. So this helps us be convinced that SCH actually does have two mechanisms of action, not only one. But is it better to have two in one, or would it be better to just treat with trimethoprim and niacin at the same time? And so to do this study, we used a population of cells that are very hardy and very difficult to kill. As you can see, at concentrations that would normally kill regular bacteria, these cells have no difference in viability than when treated with a super safe solvent. However, if you treat with SCH, you can see that these cells are still dead. And this number 10 to the second here actually means that there were no detectable cells. That's just the limit of our detection. So this is exciting because it actually helps us realize that it might be better to take one molecule that has two mechanisms of action on it rather than combining two different antibiotics. And so while SCH is a very exciting molecule, it was not created to be an antibiotic. It is originally used as a blood thinner. And so using help from a wonderful scientist at our university named Han Kim, we were able to make derivatives of this molecule that were able to increase their effectiveness. And using one of these, which we call Irresistin 16, we were actually able to show that in a Neisseria gonorrhea mouse model, IRS 16 was actually able to clear the infection when you treated these mice with a large dose of drug-resistant bacteria. So this is exciting. I hope that throughout this, wait, do I have future questions? Yes, I do. I hope that from what you've gained, I hope that what you've gained from this so far is the uniqueness of SCH, that we had to do a lot of innovative things in order to identify a molecule that doesn't become resistant. However, I want you to also realize that this work couldn't have been done by myself alone. There took a lot of great collaborators who are very intelligent and care a lot about antibiotics in order to make this project what it is. So some future questions that we still want to ask is, why is it so hard to get resistance to SCH? We only hypothesize that it's because there are two mechanisms that the bacteria have to deal with instead of just one, but as scientists, we want to test that out. Secondly, how does SCH accomplish this? Does one mechanism of action happen first and make the bacteria more susceptible to the second, or is it vice versa? These are questions that other scientists are still working on. And lastly, is this dual target mechanism of action generalizable? Can we, for example, take any of the antibiotics that I listed earlier and combine them onto the same molecule and get a molecule that is not prone to resistance, kind of like SCH? So these are really good questions that if I was to do another PhD, I would be working on, but thankfully this was enough for me to graduate. And so there are other wonderful scientists working on these questions. And so with that, I'd like to thank my advisor, Zemric Atai, and all the members of his lab, particularly Max Wilson, who helped me find SCH, and Sophia Lee, who taught me how to do metabolomics. I also want to thank Ben Bratton, who was a postdoc in our lab at the time, but now he has his own lab at Vanderbilt University. And he has done amazing work on the in vitro biochemistry that we were doing, and he was incredibly invaluable for data analysis and making the figures. I want to thank Gabriel Moore for his work on the worms, Josh Rabinowitz for letting me use their mass spec and helping me analyze the data, Joseph Sheehan, who is an incredible research scientist, and that is one of the reasons him and Ben are both co-authors with me on this paper. And then I also want to thank Han Kim for his help in designing the derivatives and helping us to identify good and correct animal models to use.